Hi, everybody. Welcome to Chapter 3. In this chapter, we're going to be going over cells and tissues. This is an extremely long chapter. Kaya, be quiet. That's my dog, one of my dogs. I think you met one, but that's another one. So we're going to start out this chapter talking about the cells. We'll focus on what are cells, how do they work in the body, what type of reactions do they do. Dudley, be quiet. That's my other dog. I don't think you met him either. I think you met Frank. And then we'll talk about why these or how these cells come together and why they come together to form the tissues of the body. The first thing we always like to start with then is talking about the cell theory. So it's a basic principle of biology, and the credit for this theory goes to Theodor Schwann, Matthias Schleiden, and Rudolf Virchow, as well as Robert Hooke, who is the first person to actually see a cell and coin the term cell. The theory basically states that all living things are composed of cells. A cell is the basic unit of life. Cells arise from pre-existing cells. So they do not come from non-living materials. Energy flow occurs within cells. Heredity information is passed from cell to cell, and all cells have the basic chemical composition. Okay. So cells are the structural unit of life. And humans have anywhere from 50 trillion to close to 100 trillion cells that make up our bodies. And that doesn't count all of the different bacteria and everything else that we have going on. Dogs don't play around. There are three basic parts to a cell: the nucleus, which you can see here, the purple structure, the cytoplasm, which is that liquid environment that contains all of our organelles, and then the plasma membrane, which is the outer barrier of the cell. So at this point, if you're following along with the study guide, you should be able to define a cell. You should be able to explain the cell theory, explain the parts of the cell theory, who came up with the cell theory. You should be able to tell me how many cells an average individual has, anywhere between 50 and 100 trillion, and the three parts of the cell. So we're going to look at all the parts of the cell, as well as talk about the functions of those different cell structures, okay? So the nucleus itself is the control center of the cell. It contains DNA, which is our genetic information, which holds the information to carry out all protein synthesis. The plasma membrane, the outer region, is a barrier that surrounds all cells. And I can say this: all cells have a plasma membrane. Some cells also have a cell wall, but in animals, we do not. So the plasma membrane keeps the internal cell contents separate from the external environment. The plasma membrane also contains cell junctions. A junction is a structure that binds cells together and allows for communication between cells. So there are multiple types of cell junctions that we'll talk about. Cell junctions help to bind the cells together. We also have glycoproteins that help to bind cells together. So here you see a tight junction. Tight junctions are junctions that hold cells together and make a leak-proof membrane between the cells. So materials can't just flow freely through between one cell and another. Desmosomes are anchoring junctions that hold a cell to another cell and they withstand mechanical pulling, so mechanical stressors. Half of a desmosome is actually called a hemidesmosome. A hemidesmosome is what holds our cells to underlying connective tissues. So when you're talking about epithelial cells, you have hemidesmosomes that hold those cells to underlying tissues. And they also prevent mechanical stress. And then we have gap junctions. So these are gap junctions. Gap junctions are communicating junctions. So they allow for chemical information to flow from one cell to another cell. I just mentioned this, but I'll go over it again so you can hear it again. Your tight junctions are your impermeable junctions. They produce a leak-proof barrier so that nothing can get through between cells. Desmosomes create or hold cells together, preventing the cells from being pulled apart due to mechanical stressors. So think about people walking next to you, bumping into you, the stress on your skin. Your skin doesn't just fall off, get sloughed off that easily. And then gap junctions. These are your communication junctions that allow for chemical information to flow from one cell to another. At this point, again, you should be able to tell me what is the function of the plasma membrane and what are the three main junctions, and how do they function? How do the three junctions function? Kind of find me there, but that's okay. All right. Now we're going to look at the cytoplasm. The cytoplasm is the material inside the plasma membrane, but outside of the nucleus. So this is an area where most metabolic activities occur. Within the cytosol, you have organelles as well as inclusion bodies. So cytosol is the fluid that suspends all of the organelles within the environment. Stop it, dogs. Stop. I have two dogs here playing right now. I have three. I've told you already, I think, that I have three dogs, but two of them are playing. Stop playing. I'm trying to teach someone right now. Apologies. Inclusions are substances that are stored, such as nutrients or potential waste products that need to be released from the cell. And then organelles are the actual machinery. They're the tiny little organs of the cell. So they're that's what I call them. They're tiny organs that function in the cell like our organs function in our body. Okay, and they all have different functions, but they work together overall. So this is a beautiful figure that shows the plasma membrane, the nucleus, your organelles out here within the cytoplasm. We're going to go over each organelle. It is your responsibility to know these organelles and how they work, specifically how they work in the body. So if there was damage to an organelle, you would be able to understand how that damage or what that damage does to the overall organism. So the first organelle, most people have heard of this one, is the mitochondria. Typically we call it the powerhouse of the cell. Mitochondria is where ATP is produced through a process known as cellular respiration. And ATP is the energy currency our cells use. Ribosomes, these are tiny little structures composed of proteins and ribosomal RNA, and they function in protein synthesis. So they're kind of the workhorse where you're producing proteins continuously. Ribosomes can be found free floating in the cytoplasm or as part of the rough endoplasmic reticulum embedded within there. So then we have endoplasmic reticulum. Endoplasmic reticulum has two types. There's rough and smooth. Rough contains ribosomes, smooth does not. So the rough endoplasmic reticulum functions in helping in synthesizing proteins and then transporting those proteins to the Golgi apparatus. And so here you see ribosome. I'm sorry. Here's a ribosome producing protein on the rough endoplasmic reticulum. That protein is then, once it's produced, is placed within a vesicle of the endoplasmic reticulum, and that vesicle is then going to move to the Golgi apparatus for further processing and for movement to wherever that protein needs to be. And here's the whole function. As proteins are synthesized, the proteins migrate into the ER. Once they migrate into the ER, they then fold into their functional form. And sometimes you may modify those proteins a little by adding a carbohydrate or an extra lipid or some other molecules. It's then packaged into the membrane of the ER, and a vesicle forms. That vesicle is then transported to the Golgi apparatus. The smooth endoplasmic reticulum lacks ribosomes, but it is continuous with the rough endoplasmic reticulum. It functions in lipid synthesis, helps in producing cholesterol and other lipid molecules. It also functions in detoxification of alcohols and other drugs. So the Golgi is the next organelle that we'll talk about. I usually think of the Golgi as the post office. So the Golgi basically takes the proteins from the endoplasmic reticulum, and then it transports them, packages them, and moves them to the proper area, and then transports them either to the extracellular environment or to the plasma membrane, or it might just make an organelle right there, like the lysosomes. And so that's why I kind of call the Golgi the post office, because the post office is where you sort materials and then you send them out, you deliver them. So in one sense, you may deliver them into a vesicle that stays within the plasma membrane. In another sense, you may deliver them to a vesicle that excretes the proteins into the extracellular environment. And in another vesicle, you may embed the proteins within the Golgi vesicle. And then when the vesicle binds with the plasma membrane, it becomes part of that plasma membrane, and the proteins are now embedded within the plasma membrane. We have peroxisomes. Peroxisomes are small sacs that contain oxidase enzymes. Oxidase breaks down free radicals. Free radicals are typically converted to hydrogen peroxide. Hydrogen peroxide is also considered poisonous to our cells, and so hydrogen peroxide is then converted to water and oxygen. Lysosomes are also sacs, organelle sacs, that contain digestive enzymes. So these break down proteins and fats and nucleic acids. Typically, you see lysosomes in or you see lysosomes as your phagocytic organelle. So they break down and dispose of materials, and then those materials can be released to the extracellular environment. They also can help in breaking down worn out materials within the cell to be reused. The cytoskeleton is a network of proteins. There are 3 types of proteins that make up the cytoskeleton, and basically these proteins function in forming a frame or a skeletal system for your cell. So it helps to maintain the cell shape. It helps in supporting organelles within the cell, and it functions in movement of materials within the cells. So there are 3 proteins that make up the cytoskeleton: microfilaments, intermediate filaments, and microtubules. And so here you can see microfilaments, intermediate filaments, and then the microtubules. Centrioles are rod-shaped bodies that are composed of multiple microtubules. The microtubules then within the centrioles form spindle fibers and help in moving DNA during replication, cell replication, and cell division. So here are the different structures that we talked about. This here is showing the plasma membrane. I haven't really talked a lot about the plasma membrane yet, but you can see the plasma membrane with the sugar, with proteins, cholesterol, and sugar pieces. I'll talk about that in a few minutes. A little bit more, I will say. Then you have the different organelles that we talked about. So these figures are right in your textbook, so you can read them in the textbook. Last thing in this lecture that I'm going to talk about are the cell extensions. So cell extensions are materials that are found in some cells, not all cells. And these are seen outside of and attached to the plasma membrane. So one type of cell extension is called cilia. Cilia are tiny little, very small finger-like structures that are attached to the cell, and they help in movement of materials along the cell surface. Flagella are longer hair-like extensions that help to move the entire cell. Microvilli are very, very small extensions of the plasma membrane that help to increase surface area for absorption. And so in the human body, cilia are located in regions like the respiratory system. They're located in the reproductive system. Flagella there's only one flagellated cell, and that is sperm. Microvilli are located in the urinary system, in the digestive system, where we're going to need increased surface area. This is a picture of a flagellated cell. This is a sperm cell. Sperm cell, just so you know, has a head which contains this acrosomal cap that contains digestive enzymes that will help to force the sperm into the egg. This mid piece, which contains multiple mitochondria, as well as some inclusions of carbohydrates. And then we have the flagella, which is the tail of the sperm. Oh, sorry. And then down here you can see cilia. So this is ciliated cell. You can see this here is pseudostratified columnar epithelium. And you can see cilia, you can see the nucleus, you can see the cell itself right here. So each of these is a cell. So at this point, you should be able to tell me what an organelle is and identify these different organelles, as well as what are cilia, what are flagella, and what are microvilli, and what are their functions. I'm going to stop here because this has been 20 minutes. I think that's long enough for a lecture. And I'll start the second lecture for chapter 3 in a few minutes. Bye.